have recovered from a cold recently, you should probably thank your T cells. Your body has an amazing defense against infections called the immune system, which is made up of many types of immune cells. We study one type of immune cell called a T cell. We are interested in how they grow and increase in number. We use a scientific instrument called a mass spectrometer, or mass spec, to study them. The first step is to create an immune response. We do this by activating the immune cells with antibodies. We are specifically interested in items called proteins that are made by the T cell during the immune response. To get to them, we break open the T cells by adding detergent. We collect the protein and wash it several times. Next, we add an enzyme to chop the proteins up to make them smaller. Then we put it in a thermomixer. This machine heats and mixes the contents. We call what we will study in the mass spec the sample. The sample is not quite ready to go into the mass spec yet. The mass spec is extremely sensitive to salt, so we need to remove salt from the sample. We are now ready to place the sample into the mass spec. After 3 to 4 hours, a single mass spec run gives information on 4,000 to 6,000 proteins. This includes the type and amount of each protein in the sample. This data is a large fraction of the proteins present in the cell. We now need to study the data collected from the mass spec. We spend a lot of time analysing the data using Excel and approaches that allow us to handle big data. These data help us to better understand how these T immune cells work. They are crucial for our body's fight against viruses and cancer cells. But if these T cells become too active, then it can result in autoimmunity. So, it is important to research how these immune cells act, including how they grow and increase in number.